Right. So do you want me to do you want to start, Shanglu, and then I'll
Yeah, I will just say a few words. So good morning, everyone. Today is Friday, our regular seminar time. Before the seminar starts, I just want to introduce a new person from my lab, Dr. Rodrigo Ramos. Rodrigo, can you show your screen? So Rodrigo just joined my lab as a postdoc. He's from Brazil. He got his bachelor, master, and PhD in Universidade Federal de Viçosa. So he had 3 years of postdoc experience. Then he came here just 2 weeks ago. So welcome, Rodrigo.
Thank you, Dr. Shanglu, for the introduction, and thank you, everyone. So hopefully have a great time here and contribute as well to the seminar and some of our knowledge background back in Brazil, and bring some positive contributions to the program. So thank you.
Okay, well, good. So now, Dina, you can introduce Glenn.
All right, so bem-vindo, Rodrigo.
Yeah, thank you.
Francisco, we have a Portuguese-speaking constituency here that will keep you busy. We'll get me speaking Portuguese again. Okay.
Yeah, but Dina has a very rusty Portuguese. She has to practice before talking to us.
Yeah, but at least I speak Portuguese. All right, let's not start that. So, all right, so today's seminar speaker, is my honor to introduce Dr. Glenn Scholes. I'm going to give you a quick, very quick introduction, because if I were to tell you everything he's done, I would be here for a while. So Dr. Scholes got his bachelor's from Cornell, and then he did what I know many of you graduate students, and so did I, did for a bit, is be a technician. He was a medical and veterinary entomology technician, first at Cornell as a field supervisor for onchocerciasis work. And if Jack Peterson is in the audience, he'll relate to this. So he was in Liberia, West Africa, actually also for the Johns Hopkins University. Then he was back in Cornell working as a lead technician in the insect chemical ecology research lab. He then got a master's and PhD vector biology from University of Notre Dame with Dr. George Craig. He was working on Aedes albopictus, on mosquitoes, and vector and their transmission of canine, of dog heartworm. Then he did a postdoc at Plum Island, and if Jim is in the audience, Jim Mosey, you'll be really interested to hear this, at the Animal Disease Center, working on African swine fever virus. And then at the Vector Bioecology Lab at Yale University, working on Ixodes scapularis borne pathogens. In fact, he discovered Borrelia miyamotoi and was the recipient of a Yale Brown Cox Fellowship. In 2000, he joined USDA-ARS Animal Disease Research Unit in Pullman, Washington, and he has been a research entomologist there. And I think he's going to talk to us a lot, and he's told me that, about the work he's done there on mostly Theileriosis, but also work on babesiosis, anaplasmosis, and equine paraplasmosis. He then just recently, so in 2019, came back to the East Coast, where he's been running a program at USDA-ARS Invasive Insect Biocontrol and Behavior Lab in Beltsville, where particularly of interest to us is developing a new program called Ticks and Human Health. Many of you are going to have the opportunity to talk to Dr. Scholes, and I'm really interested to hear his seminar. So Glenn, take it away.
All right, am I on screen? So thank you, Dina, for the introduction, and it's my pleasure to be here today. And Dina and I have been talking for a long time about me coming up to Rutgers to give a talk. And in fact, it was last year, about the one day before they shut us down, that Dina was at my lab visiting and talking, and my seminar was canceled. And so now this is the rescheduled seminar 1 year later. And so for many years before I moved to Beltsville, research in my lab focused on Tyleria of livestock. And so today I wanted to share some of my past research experiences and findings with you in hopes that the work with Tyleria that I did out in Pullman would help inform our response to the Asian longhorn tick, which you here at Rutgers or there at Rutgers have so much experience with in New Jersey. So before I get started though, I'd like to share a little background with you about the Invasive Insect Biocontrol and Behavior Lab where I currently work. The mission of the IIBDL, as we call it, is there on the slide, is to conduct research on insect pests of agriculture and public health and mitigate other threats to our nation's supply of food and fiber. But as you can see also, one of our newest project plans is focused on ticks and tick-borne pathogens in humans, which is the primary reason I've relocated here after nearly 20 years working in livestock pathogens out in Washington State. So why did I really relocate to Beltsville after spending so much time of my USDA career working on livestock disease in Pullman? Well, in Pullman, I was the only entomologist in the unit and made up mostly of molecular biologists and veterinarians. And that kept my area of expertise in demand, but it was always difficult to do my own work and while also supporting other projects. So I ran the tick lab there. I was a collaborator in both tick-borne disease projects. So we worked on a wide variety of cattle, horse, and sheep diseases that are transmitted by ticks. But in November of 2017, which was shortly after the longhorn tick was discovered in New Jersey there, I was approached about the possibility of transfer to IIBDL. The ARS had envisioned a programmatic shift to focus more research effort on the problem of human biting ticks. And since my postdoctoral experience was with Enzootic scapularis, it was thought that I might be interested in transitioning back into ticks and human health. So I arrived in Beltsville to start that work in August of 2019. So you can see that the wheels of the USDA grind slowly when it comes to these kind of things. It took almost 2 years for my transfer to become complete. And I was still trying to get things set up and running when the pandemic shut us all down, March 2020 or March 2019. That's a mistake. Anyway, so the new project plan was initiated about 3 months after my arrival, and I'd helped to write this plan, but since I wasn't present, I wasn't listed as the project lead at the time. But I did help to develop this plan, and you can see from this plan, it's focused broadly on ticks of critical importance for people in northeastern United States. And the tick of primarily concerned with obviously Enzootic scapularis, but we also have in here focus on Dermis variabilis and Glioma americanum and the newly invasive longhorn tick, which is the main reason I wanted to talk to you folks at Rutgers to see if I could build some collaborative relationships on the longhorn tick. And as you can see, all 3 pillars of the project plan are surveillance of both ticks and pathogens, control of ticks, and understanding the roles these ticks play in disease transmission. So the surveillance, control, and disease transmission are our 3 pillars. So just wanted to sort of quickly, briefly talk about a few of the things that I'm working on before I dive into my historical talk, which is most of the data I'll be presenting today is from past work out in Pullman. But because of the need to renovate lab spaces and hire technical help, and because of the pandemic and all, it was a little bit of a slow start. So since my arrival here, I've been continued to work on projects connected to ADRU, mostly centering around cattle fever tick and grant-funded projects that I still have out there. But I've been able to get a few things going here, and I hope I'll be able to come back to Rutgers at some point or talk to some of you during this visit about some of these projects as well. I've already had some nice interactions with Dina and others on some of these projects. But so 3 or 4 things that I've gotten started so far are methods for detecting ticks in soil samples, using artificial mouse nest boxes as a tool for tick monitoring and delivery of host-targeted control strategies, strain typing of Anaplasma phagocytophilum in ticks, mice, and deer, and biological control of ticks using hymenoctrin parasitoids. So none of these are things that I have enough information or data to talk to you about today. And I thought that my past experience working with Tyleria transmitted by ticks in the US would mesh well with the expertise that is present at Rutgers on the longhorn tick. And maybe we could take some of my background on this tick as a potential risk to livestock and add it to some of your expertise on the longhorn tick and come up with some interesting projects, or at least that some of my work could inform the sense or the feeling of what the risk of the longhorn tick might ultimately end up being. So back to review a body of my previous work on tick-borne pathogens of livestock at ADRU. And specifically, I'd like to talk about work with Tyleria species that are infecting cattle and horses and ruminants worldwide. So we have Tyleria of cattle in the United States. There's no real problems with these right now other than silent infections, which we'll talk about in a minute. But worldwide, there's Tyleria parva, Tyleria annulatus, and Tyleria orientalis in Asia and the Pacific Rim, which is being transmitted by the longhorn tick there. And then I've done quite a lot of work on equine Tyleria, which has been eradicated from the United States, and we'll talk a little bit more about that. There's also Tyleria of small ruminants, which have a worldwide effect, especially on smallholder agriculture, and can have devastating effects on sheep and goat herds. So in the United States, species of livestock Tyleria are all either considered exotic or they're benign. And let me explain what that means. So exotic we would classify as something that we do not have in the United States, and our main primary risk is external. Equine Theraplasmosis, for example, is considered an exotic Tyleria in the United States. We're one of 9 countries in the world which does not have this pathogen. And that's because it was eradicated from the United States through an eradication program. And there's US regulatory controls on importation. But this pathogen can cause persistent infection in carriers, silent persistent infection in carriers. And so it's the subject of eradication efforts continuing. And then by benign, I mean there are several species of bovine tyleriosis, including Tyleria orientalis, and others that are now classified within the orientalis group. These occur sporadically. They're found usually with asymptomatic cattle back as early as 1950, but some of those were identified without any molecular tools, so it's kind of hard to know exactly what it was they found. More recently, 1993 and 1997, we were importing Wagyu beef breeding stock from Japan, coming from an endemic zone for Tyleria orientalis, and none of those cattle were tested. So we presume that some of those were infected. Japan stopped allowing importation of Wagyu beef in 1997 because they felt like it was impacting their market, their ability to sell the Wagyu beef out to us if we could grow our own. So, but anyway, that may have been a source of importation. These bovine Tyleria are usually considered benign, and they're not currently under any regulatory control. And then I've worked for many years off and on since 2005 on Tyleria parva, which is not of any risk to the US cattle populations. It's very important in Africa, but some of the things we've learned from working with Tyleria parva may be informative in terms of the risks of Tyleria in general in the United States. So Asian longhorn tick was introduced in the United States, obviously, and it changes everything, right? The Asian longhorn tick brings with it the risk that it will transmit exotic Tyleria species that are currently present and circulating silently in the US. Longhorn tick is reported as a vector of orientalis throughout Asia and the Pacific Rim area. It's also reported to be a competent vector of Tyleria equi in lab model studies, although it has not been tested in actual transmission trials. Previously, the maintenance of Tyleria species in the US relied only on inefficient vectors that might be present here, or iatrogenic transmission, or mechanical or transplacental transmission. So these are low-level silent infections. Now, with the presence of the Asian longhorn tick, there's the possibility of efficient vector-borne transmission. And that leads to this key concept I need to really emphasize, that the severity of disease caused by Tyleria infection is dose-dependent. So the more the dose, the higher the dose, the more likely the animal is to succumb to disease. So we can have inefficient transmission, which is what I talked about here under the previous situation, where Tyleria infection is there, but it's transmitted inefficiently, and the animals are not showing any signs of disease. Whereas if we add that now, and there's efficient transmission by competent vectors, this will result in high levels of infection, high levels of transmission, which could lead to disease. So the presence of the Asian longhorn tick changes the whole equation for these silent Theileria in the United States. And you all know the history of the longhorn ticks, and this is actually map is a little bit out of date. I don't need to tell people at Rutgers about the Asian longhorn tick probably, but it was first identified by Rutgers in August 2017. And you can see that it's spread through this zone since then. And this map is actually a little bit out of date, but the point is that this tick is present and spreading, and it doesn't seem to be involving itself so much in transmission, possibly in transmission of human disease, because it doesn't seem to like to feed on humans. But there's definitely been cases in Virginia here where we think it's involving itself in transmission of Tyleria orientalis. So let me look at the Tyleria life cycle briefly for you and show you that some of these pathogens that I've been working with over the years. And first of all, Tyleria is an apicomplexan protozoan. So for those of you who don't know what that means, the life cycle is similar to malaria or babesia. The tick is the definitive host. This is an important idea and something that parasitologists appreciate. The parasite life cycles between the tick and the vertebrate host, but the sexual stages of development occur in the tick. That's where the diploid stage is. Asexual development occurs in the vertebrate host, haploid. So that's the same as for malaria. So as in malaria, the vector is the definitive host of this pathogen, and the pathogen cannot complete its life cycle without passage through the vector. So Tyleria has specific hallmarks that make it different than other babesia or other apicomplexan. Sporozoites are transmitted in the saliva of the tick, and those sporozoites invade and replicate in leukocytes, or peripheral blood mononuclear cells, also known as white blood cells. And those merozoites, the sporozoites develop into merozoites in the leukocytes and are released to invade red blood cells, which is the red blood cell is the tick infectious stage. So the tick has to pick up red blood cells infected with the Tyleria in order for the parasite to continue developing in the tick. This is in contrast to babesia, where the sporozoites invade the red blood cells directly. So there's three pathogens we're going to talk about today: Tyleria equi and Tyleria parva are two extremes. So Tyleria equi undergoes a single round of replication in the peripheral blood mononuclear cells before release of merozoites in red blood cells. The primary pathogenesis is due to anemia, and those tick red cells, or those red cells that are parasitized, are destroyed, and the animal suffers from anemia. Tyleria parva is all the way at the other end of the spectrum in terms of pathogenesis. It has multiple rounds of replication in peripheral blood mononuclear cells, and merozoites are released and go into red blood cells, which is a dead end in the host, and those are picked up by ticks. But the peripheral blood mononuclear cells that are infected by the merozoites are actually transformed, is the term they use. It's very similar to almost like a cancer. The cells replicate rapidly, and the replication of the peripheral blood mononuclear cells is linked to the replication of the parasite, and pathogenesis is due to this lymphoproliferative disease. So the animal basically drowns, its lungs fill with lymphocytes, and it drowns basically in its own lymphocyte populations. It's increased very rapidly. Tyleria orientalis, which is what we find here sporadically in the United States, is sort of between those two. There may be has similarities to both Tyleria equi and Tyleria parva. There's multiple rounds of replication in the peripheral blood mononuclear cells, but there's no transformation. And pathogenesis in this case is due to anemia as well. So how can previous research on tick-borne Tyleria inform our response to the longhorn tick? In my lab, we've studied the vector biology of equine Theraplasmosis in terms of response to outbreaks, vectors, treatments, eradication of outbreak. The vast majority of my work was done has been done with Tyleria equi, but some of this work also led to identification of a new, previously unknown species, Tyleria haemaphysalis. And we'll talk about that some more in detail later. Then I did work on East Coast fever, Tyleria parva, and that work was based on studies of acquisition and transmission from low-level persistent carriers. And I think there's some information you can glean from that to inform what might be happening with the longhorn tick. And then finally, when Orientalis showed up, we were involved in identification of infection in asymptomatic cattle by testing blood samples. But we also were early partners in that Virginia outbreak. We helped to isolate the pathogen and made cryopreserved stabilites of that for use in laboratory infections so that we could study the transmission in ticks. And thanks to Dana Price, we were able to get some ticks from New Jersey and start our own colony of longicornis out there in Pullman, and that colony has been used and will continue to be used for transmission studies with Tyleria parva and other livestock pathogens that the tick might become involved in. I guess I'd like to say here too now, if anybody has questions or if I misspeak, please don't hesitate to interrupt me. Equine Theraplasmosis, let's start there. I spent a lot of time working on equine Theraplasmosis. It's an enteroerythrocytic protozoan, as I've said. Obligate host is the tick. It infects equines. That includes horses, donkeys. It's common in zebra in Africa, and then it hybrids as well. So mules are susceptible. And there's places in the literature where it mentions infection of camels. However, that may be a misidentification or another species is very closely related. The disease it causes, of course, is referred to as equine Theraplasmosis. Acute infection is characterized by fever, depression, anemia. Case fatality rates as high as 15% have been reported, but that seems to be the high mortality rate seems to be a phenomenon of places where equines are under stress. And in the United States, the case fatality rate is considerably lower than that because our horses are much better cared for. So they don't have other comorbidities. This infection resolves to a long-term chronic infection in the animal. If the animal survives the acute infection, it resolves to a long-term chronic infection with no clinical signs. So chronic carriers can be around and may be difficult to detect, and those can serve as carriers, as reservoirs for infection. So let's go look at the history of equine Theraplasmosis in the United States. It was first recorded here in Florida in the 1960s. Not really sure exactly how it got here, but there was a joint state and federal control program started to eradicate this pathogen. The first serological assay was developed in 1970, complement fixation. Up until that time, the only way to detect infection was by clinical signs, which obviously some of them sometimes are silent, and by looking at blood smears. But blood stage parasitemia is very low, so it's not always detectable. Or by taking blood from an infected horse and injecting it into a splenectomized horse and watching for signs of disease or pathogenesis in the splenectomized horse. That's a pretty cumbersome way of diagnosing a horse, and it has to be a pretty high value animal to justify that effort. This disease, though, using the tools available and the tests available at the time, was eradicated from the US, and the US was declared free of these equi by 1988, after about 25 years and $12 million, which was a lot more money back then than it is now. Sporadic cases, though, have occurred, and I'm going to show you a map of some of those cases since 1988. These mostly are import related because the complement fixation test, which was still used as the import test for up until 2005, produces false negative tests occasionally, difficulty detecting early infections. So there's no real strong evidence for endemic tick-borne transmission in the United States since it was eradicated. But in 2005, a new regulatory ELISA assay was introduced, which has high sensitivity. And this more sensitive assay is better for keeping infections out of the United States. However, we probably let lots of persistent carriers into the country in the interim time. So let's fast forward a little bit to 2008. There was an outbreak in Florida. There was 20 horses infected. This was in quarter horses that were involved in unsanctioned racing activities. Unsanctioned, another polite word for illegal, because apparently these were cocaine dealers with more money than they knew what to do with, and they were buying and selling horses and racing them in stakes races and so forth. And so when this was discovered, these horses seemed to have been infected all through this method called blood doping. They would take blood from one animal that was supposed to be a good runner or whatever and infect it into another animal just before the race to give it more red blood cells, more oxygen carrying capacity supposedly, and help it to race. So this is clearly unsanctioned illegal activity. And there were 20 horses that turned out to be infected, acquired infection in this way. And they were all euthanized. And this was possible because these people didn't want any questions asked. You tell them their horse is positive, they don't want you snooping around, so they just put it down. And it was fairly straightforward to eradicate this outbreak. But there didn't appear to be any tick-borne transmission in this outbreak. But the next year, we found a new outbreak. And we don't know that there was any connection between this and the Florida outbreak. This possibly was a much longer standing, but then also probably related to horses imported prior to 2005 when the new ELISA was developed. This was the King Ranch in South Texas. King Ranch is famous as a cattle ranch, but one of their biggest businesses of the last many years is producing registered quarter horses. So they found an index case was infected, and then that led them to test the whole herd. And they found that 290 horses were positive on the ranch. 81% of the horses on the ranch were positive. It was 100% infection in 3 of the 4 non-contiguous ranch divisions. And only young stock in the breeding herd were uninfected. So that breeding herd was one of the divisions where that's where the lower rate was. So in this case, there were many very high value animals, and this was a well-known and wealthy Texas family who owned this. And many of these horses were what we would refer to as the foundation genetics for the quarter horse breed. So euthanasia is not considered a control option for at least some of these cases. The ranch was placed under quarantine, internal management and treatment programs were developed, and epidemiologic trace outs were done to find any horses sold off the ranch. There were trace outs actually to 17 states because the ranch was selling quarter horses all over. We'll talk about that in just a minute. My job here then was to look at vector competence of Texas ticks. Were we having tick-borne transmission of equine Theraplasmosis? Well, there's 30 exotic tick species and 6 genera that are competent vectors, and that includes Infectious longicornis in lab model of transmission. And this was well before longicornis arrived here, so the transmission was probably being involved local species of ticks. And we know that Rhipicephalus guaphalis micropus, the cattle fever tick, is a highly competent vector, and that may have been present at one time or another down in the ranch. And then Dermacentor variabilis is also a competent vector. So in response to the outbreak, we checked the horses on the ranch for ticks and tested several tick species native to South Texas for vector competence. We collected ticks from 228 horses on the ranch, and you can see the color coding here. We tested, the green ones are ones that were competent but inefficient vectors, and the red ones are ones that were highly efficient vectors. So we tested Amblyomma caginense, which is the genus has now been revised, so it's now known as Amblyomma mixtum. 79% of horses had caginensis. 20% had Amblyomma maculatum, the Gulf Coast tick. 16% had Dermacentor variabilis, the American dog tick. And 3% had Dermacentor nitens, which is also known as the tropical horse tick. We tested these species by placing them from infected horses onto uninfected horses, and were able to transmit with Amblyomma caginense and Dermacentor variabilis. Then we tested some colonized ticks as well. We tested a colony of Amblyomma americanum, Amblyomma imitator, which is very closely related to mixtum, Cajunense mixtum. And we tested Amblyomma maculatum from a colony. And we also tested Boophilus, Streptocephalus, Boophilus micropus. And turned out that micropus was a highly competent vector of the Theraplasmosis. So you can see from this map that trace outs of horses sold from the ranch. There's 412 cases total in this outbreak that could epidemiologically be linked to the ranch. 16 states, and you can see the distribution of those here. All positive animals were either quarantined in situ under supervision of a state veterinarian, which was a rare thing because it's expensive. They were returned to the origin ranch. The King Ranch had voluntarily agreed to take back any horses that came from their ranch that were infected. Those horses on the King Ranch were all enrolled in an experimental treatment program, which we developed at the Animal Disease Research Unit in Pullman. Any animals that weren't enrolled were euthanized. But you can see here, this is a different map of the cases, 124 cases that could not be linked epidemiologically to the outbreak. These are cases that were present in the United States, and we have no epidemiological reason to suggest they came from the King Ranch. And so it's just cases that exist silently in the US and were discovered only because we were tracing out from the outbreak. These were here, and it's assumed that these represent a persistent low-level infection population of horses in the United States that are persistently low-level infected. So these persistent infected horses, obviously, as I said before, can be reservoirs for pathogens. Pathogens dose of the level of infection has an effect on vector competence, of course, and some of these were probably there was low-level transmission going on or whatever, but the vector is the definitive host with Tyleria, and so it takes a very close association between tick and the parasite pathogen for these to actually be transmitted. The Florida outbreak, all the transmission was iatrogenic, so we don't think there was any bloodborne transmission. But the Texas outbreak was so large and of such long standing that we presumed there was tick-borne transmission going on. So the take-home point from this is that knowledge of the competent tick vectors in the area is essential to understanding the risk of establishment of endemic transmission. And I'm going to make the point later on that I think that the presence of Haemaphysalis longicornis in the United States, coupled with this presence of these persistently low-level infected horses, may be a risk factor that we definitely need to pay attention to. So the resolution of the outbreak, the treatment program was developed in our lab by some of the veterinarians I worked with, and 162 horses were treated and cleared of infection. This clearance was defined by actually pooling blood from a group of horses and injecting it into splenectomized animals to confirm that they were truly cleared. But unfortunately, the cleared animals, the drug-treated animals that were cleared of infection, continued to be seropositive for several years. So those were what we call regulatory positive, and that was a regulatory issue. And I'll bring it up in just a minute. At the time of the outbreak, the treatment was very expensive because the drug, imidocarb dipropionate, is formulated for dogs, for use in dogs, and there was no livestock formulation of this drug. So they were using a dog-formulated drug to treat horses off-label, and the small vials that were available for treating dogs weren't sufficient for a horse. They had to use many, many vials to treat a horse because of the large blood volume. And so the outcome, the consequence is that treating animals was very, very expensive. And so any horse that wasn't valuable in some way for genetic purposes or as a racing stock or something had to be euthanized. So we're able to save 162 of those 400 horses that were actually, 400 plus horses that were actually in the outbreak. Some animals that were in the outbreak are still seropositive, and they have to remain quarantined by statute because that's regulatory positive. Regulatory positive means they test with a approved regulatory assay as if they were infected, even though we can confirm they're not infected by sub-inoculation. They still have to remain quarantined. So parts of the range of beasts now continue to be quarantined. And I put this picture in the background of a horse with snow on it because of the topical nature. The King Ranch experienced this winter cold snowstorm. This was 4th of February 2011. So you see all the news about the winter storms in Texas. You know, it's not an uncommon thing. So Texas gets snow and rain sometimes. Just an aside. So as a part of the process of finding and working on Tyleria equi, we discovered a new pathogen, Tyleria haemii, which was in horses as well. There was a veterinarian down at the border, worked for the USDA, named Daryl Haney, who was checking horses that strayed across the border, and he did a blood smear on a horse and was lucky enough to find what appeared to be a positive. However, that animal was serologically negative by complement fixation, and blood passage. He sent us blood, and we had to pass his blood to a splenectomized animal to confirm that it was actually infected. And it was infected with an uncharacterized protozoan. So we did sequence the 18S ribosomal RNA and did an analysis of all the sequences in GenBank and showed that this was a separate, uniquely different group. And you can see, so these are up here, the yellow at the top is all of the horses that were from the outbreak, or a sample of the horses from the outbreak. And this group down here ties back phylogenetically to zebra isolate from South Africa. So this haemii was very distinctly genetically different. And we had developed the microsatellite markers for doing population genetics of these outbreak horses. And this was distant enough that it could not be typed with that same microsatellite array. So we published the paper on the microsatellite markers and the identification of this as a new species. And then just recently, there was a genome sequence and a description of this as a new species published in 2018. So Tyleria haemii is here as well, and possibly circulating silently in the US. East Coast fever, I wanted to bring this work up because it informs us about the ability to detect low-level infections and the ability of low-level infections to be transmitted by ticks. But the United States is not at any risk from Tyleria parva. This is just information from this parasite that can inform our choices about how we approach Tyleria that are in the United States. So this is a pathogen that is common in large parts of Africa. It's a very devastating disease. It kills the cattle. It's a very high mortality rate. It replicates in lymphocytes, as I said before, and ticks. The tick is the definitive host, as before. Rhipicephalus appendiculatus, that's the brown ear tick, is the common name of that tick, the primary vector in Africa. And transmission is strictly transstadial, so nymphs acquire and adults transmit. The pathogen poses no current risk to the US, as I said, but we can learn a great deal about Tyleria from studying its vector biology and pathogens. So just quickly, the Tyleria life cycle is very similar, and the key is that the animals do not die from anemia as they do with Orientalis or equi. They die from lymphocytic proliferation, which ends up causing a pneumonia-like syndrome and ultimately results in death. So tick acquisition and transmission from persistently infected cattle in this disease is never been studied. It's because it's very difficult to study a pathogen which is in such low levels that transmission is very hard to demonstrate. And so studies in the lab are always done with acute infections, which are easy to study because you have a lot of parasite you can see. But acute infections are not epidemiologically very meaningful because the animals mostly die. And so those animals don't actually survive long enough for ticks to feed and transmit from them. An animal can be dead in 9 days. So it's a very devastating, quick-acting disease, but only studied in the acute infection. And so we wanted to, in my lab, study persistent infection because we believe it had more epidemiological relevance. But it's a very hard experiment to do. And so it took some effort. East Coast fever is controlled in Africa mostly by tick control, which is expensive. But the problem of wildlife host sustaining tick populations is it's very revealing if you go to Africa and you stand up on a hill and you see a herd of zebras and a bunch of giraffes and other things all out there intermingling with the cattle. You know that the same ticks are feeding on all of them. So it's very difficult to control a tick population in that kind of setting. And there's also health and safety and environmental concerns with chemical tick control. So this vaccine was developed. It's a live attenuated vaccine. And it's difficult to produce because the live sporozoites have to be produced in ticks. And then those sporozoites are isolated and administered to cattle, along with tetracycline to attenuate the infection. So the animal gets an infection and it survives and remains a low-level persistent infection with protection from disease. This is very expensive and difficult because it requires a cold chain. The sporozoites have to be kept frozen. It's difficult to produce. It comes from feeding ticks on infected cattle and dissecting ticks and isolating sporozoites. And it may or may not, this is a matter of debate, result in a carrier state, which could bring the disease into uninfected areas. And so there was a limited deployment of this, but it's the only effective vaccine at this point. And that vaccine is produced by taking cattle at this acute stage of infection. This is the parasitemia. This is just a schematic. So, but the parasitemia gets very high initially, and that vaccine is produced by feeding ticks at this point in the parasitemia. Any of these animals that survive, few do, but any that survive, develop this long-lasting low-level parasitemia. And this transmission of this pathogen has never been studied at this stage of infection, only in this acute peak. So endemic stability is a concept I wanted to bring in here because these cattle get this low-level persistent infection, and in that state, they're protected from disease. They are infected, but they're protected from disease if they're exposed to other isolates or stresses of the kind. So endemic stability is it's known as a state in which the force of transmission is high. So animals all get infected very early, and they're infected in an early age where they're young and disease is mild compared to when they're older, infected, naive animals. Older, naive animals would die immediately. So the prevalence of disease in older age classes is reduced by high levels of protective immunity acquired when they're young and these low-level infections. Endemic stability can be established naturally by any pathogen which confers immunity from protective immunity from infection. And in some cases, this is a really interesting concept. It's been described as an ecosystem service in parts of Africa where there's other pathogens, including pathogens related to Tyleria orientalis, which infect cattle that don't cause disease and may provide protection. So my ultimate goal, if I were still working on this, would have been to investigate the possibility of using Tyleria orientalis or related non-pathogenic Tyleria as vaccination approach. The paradox here is that control measures that reduce the force of transmission can counterintuitively result in an increase in clinical disease. So if you control the ticks, young animals don't get infected. They're naive when they're older and more susceptible. They get infected and they die. So disease outbreaks are the result of a disruption in endemic stability. And this was the case in the United States for cattle fever as well. And that's why we had to eradicate the cattle fever tick in the United States. And I won't go into that story now, but I'm happy to talk with any of you about it if you're interested. This work was done by my postdoc, Cassandra Holes, who's now on the faculty at Kansas State University. And she was very brave. She's from South Africa, so she's brave anyway. But she was very brave to take on a study which was something that had never been done before and had very little chance of success, because but we wanted to be able to say something about the ability of ticks to acquire and transmit parvo from these low-level persistent infections. So we thought that understanding the dynamics of persistent transmission would be the key to understanding maintenance of endemic stability in the field. This has been published 2018. Interesting paper if you want to go look at it. Just really briefly, the strategy was to infect cattle with a pathogen using the infection treatment method. So we attenuated the infection and they survived. And we used two strains. We used one that was known as a persistent infectious strain, and we used one that was thought not to persist. So the Merrick-Cabrini strain and the Maguga strain. And we kept those cattle for 4 months, testing them along the way. And you can see that we were able to detect infection out to about 55 days here. Much more difficult to detect Maguga infection. But these were undetectable infections by the time we got to 4 months. We fed 500 ticks on them at that point, 500 nymphal ticks. And those 500 ticks, when they engorged, they molt to adults, and we took a sample of those adult ticks to evaluate for infection, and we took another sample of them to transmission feed on 2 other cattle. And the transmission feeding was done with one group of 20 ticks, so a low-level transmission, a low number of ticks, and one group of 200 ticks, so it's a higher order of magnitude higher level of transmission for both these Merrick-Cabrini and Maguga strains. One which was thought to be persistent, one which was thought to be clear, but both of these cattle were not positive for infection by PCR when the ticks were fed. When we evaluated these ticks for infection, we used a sample of those that we used for transmission. We found that only one tick was culture positive in this group, and in these we found no ticks positive by PCR or culture. However, so this just describes some of the detection techniques, and I think in the interest of time, I'll just breeze past that and show you the results, which are, this is the Merrick-Cabrini animal, and that's the one that was presumed to be persistent, and this calf received an infection from those ticks. Remember, it was undetectable or very poorly detectable in the ticks. This was 20 ticks, this was 200 ticks, and we were able to transmit and show these animals were infected. And this was there are 2 different assays here. One was a PCR assay on lymphocytes, isolated lymphocytes. One was a dot blot assay on 18S. And so we had positives with both assays. The red arrow is the dot blot positive, and the red dot is the PCR positive. So we got transmission with the presumed positive Merrick-Cabrini isolate, but we also got positive with the Maguga isolate, which was not supposed to be persistent. So ticks fed on a 4-month post-infection animal that seemed to be completely clinically clear, they were able to transmit. So this tells us, and I think this is where we can learn something applicable to transmission of Tyleria by other tick species, is that the transmission from persistent animals is possible, and it can be silent. So persistently infected cattle do not need detectable levels of parastemia to be infectious to ticks. And tick-borne transmission does not necessarily result in acute disease. But remember, this was 200 ticks, and they were very low level infected. If you throw in the Haemaphysalis longicornis, some of the numbers of ticks we see feeding there in thousands, we're talking about a greatly increased dose. So if Tyleria orientalis behaves in the same way as parva, we think that low-level infections are going to be a source of infection. So, normal studies would grossly underestimate the infection rate, but we use these newly developed, very sensitive detection assays, and we're able to show that we got transmission. So now I think epidemiological studies need to confirm this at some point, but since I've left this behind, I don't know who is going to follow up on that. Hopefully someone. So let's go into Tyleria orientalis. Tyleria orientalis is a species complex.
There's numerous bovine Tyleria species and strains that are present in the United States. Most of them are considered part of this Tyleria orientalis complex, and it's very difficult to really understand the relationships here because there's 11 members worldwide so far. Reported from the United States, starting as early as 1950, these were reported from smears and other clinical disease. So some of these we don't really know much about. This one was referred to as Tyleria buffali, which would currently now be considered part of the orientalis complex. In Michigan and Missouri, Tyleria mutans, which is also part of this complex, from Texas. There were also unspecified or uncharacterized Tyleria detected in cattle in Arkansas, North Carolina, Oklahoma, and Texas. And then in my own work, we had a set of cattle from Texas, border stray cattle, we call them, and a routine set of samples from Texas A&M that we were using as comparison group to those border strays. We initially thought that some of these cattle straying across the border were Tyleria positive, but let's put an update in there since this is an older slide. But we couldn't confirm Tyleria in any of those border stray cattle. But in cattle from Texas A&M University, they were submitted to Texas A&M as routine samples for nothing related to Tyleria for other diagnostic purposes. We tested them for Tyleria, and we found 3 out of I think it was 98 total that they were positive for Tyleria. One of those happened to be a Wagyu animal, which you recall what I said about the Wagyu beef before. 2 of them were Angus, and they have been typed as the Kytos genotype, which is different from the genotype that was identified in Virginia. The Virginia outbreak with Tyleria orientalis was typed as the Acute genotype. And we were able to, as I said before, cryopreserve specimen or blood from that outbreak and use that for laboratory infection trials at Animal Disease Research in Pullman. So here's another map of that distribution of longicornis. Again, this is actually from the newest rep, so it's more up to date. This is the Albemarle County area where the original Tyleria orientalis infected calf was found. And that was first publication. They just found a calf, but they weren't able to tie it into orientalis. But the reason that they involved the lab in Pullman where I work was because they knew that orientalis was implicated as having been transmitted by longicornis in Asia, and they looked for orientalis in anemic cattle that people had said were dying because they had anaplasma. But they chose to look for orientalis because they suspected there might be some relationship between orientalis being transmitted by longicornis. And so it turned out they found orientalis before they actually were able to implicate orientalis in transmission, or longicornis in transmission of orientalis. But then subsequently, field-collected orientalis field-collected longicornis were found that were infected with orientalis. And then this paper is in review right now. It's in revision, but preprints available. And then we were able to use that colony of ticks that we had from Dana and do a transmission trial and show that longicornis was able to transmit the isolate from Albemarle County. And so it's competent vector for Tyleria orientalis. These stars represent the other states where these uncharacterized other species of Tyleria have been found. And so I don't think it's a stretch to imagine longicornis spreading to these areas or even encountering other orientalis that we know nothing about. So I think that this is likely to be a big risk factor going forward. And so looking at how our previous research on tick-borne Tyleria might inform our response to longicornis. So just review a few of these things. There's undetected silent cases of equine and bovine Tyleria present in the US. These include the previously known and unknown uncharacterized species. They're very hard to detect based on our work with parva and also work with equine, which I didn't talk about. So we know these cattle are out there, but you have to do a very extensive workup to actually show that the cattle are infected. Cattle and horses with persistent Tyleria infections are potential reservoirs for tick-borne transmission. And We confirmed that with Tyleria equine research, as well as with Tyleria parva supports that hypothesis as well. And then thinking the idea of vector competence versus vector capacity. So there are ticks that are competent to transmit Tyleria or equine that are present in the United States, but they have a very low vector capacity because transmission is inefficient and the numbers that feed are low. There have been no studies on indigenous vectors of Tyleria orientalis, but if we were to imagine large numbers of longicornis feeding on these persistently infected animals, and sometimes those numbers can be very, very, very high, that's going to really crank up the dose of transmission, which may be enough to result in disease. Longicornis is known to transmit orientalis in the Pacific Rim in Australia, but vector capacity of this invasive population has just been established in that paper I showed you. So we know that it's competent to transmit the locus, the species that are present here as well, or to be transmitted by but we don't know anything about other species that might be retained, but we do know that longicornis is potentially a vector. And for equi as well, that study still needs to be done, but there's a study from Japan reporting equi transmissible by longicornis in a laboratory model. So all the pieces of the puzzle are present, and the body of evidence suggests that invasive Asian longhorn tick will become a vector of endemic or exotic Tyleria, which are endemic in the United States, such as the Tyleria equi, isolets that I pointed out in that earlier work, or the Tyleria orientalis infections. So I'm running short on time here, and I just want to put up these little points for discussion. Endemic exotic Tyleria are currently circulating silently. How are they maintained without vectors? Probably by iatrogenic transmission. But we think that dose is a critical factor with Tyleria, and if the large numbers of longicornis transmit, we may end up seeing transmission and disease. Premonition is the concept that infection can produce resistance to disease, and so we wonder if there might be premonition occurring in some of these populations of cattle, and would they be protected from severe disease? We don't know that. And then, of course, there's always the possibility that endemic stability could develop if this progressed further. But I think the biggest question for me is, will the Asian longhorn tick continue to expand this range south into Texas? And if so, will it involve the second transmission of other bovine and equine babesia as well, both babesia bovis, babesia bigemina, and babesia cloth? And with that, I'll stop and take questions. And this work was done over about more than a 10-year span, so lots of people involved. You can see all of the different possible collaborators and so forth there on the list. And thank you. Happy to take questions.
Thank you, Glenn. Questions for Glenn? All right, we have just a few minutes.
Okay, so if nobody's stepping, I mean, a lot of the students are going to have a following hour to talk to you. So, wow, this was kind of a whirlwind of information, especially the stuff about what you're calling endemic stability, which is kind of in the human malaria is acquired immunity. Is this sort of, you have the infection, and if you survive it, you then maintain low levels. And you're basically saying that the expectation was that, I'm just trying to wrap my head around all this, is that the expectation in Theileria was that it was not transmissible to ticks, but you've demonstrated that even years, and I wasn't clear what the time scale.
So the idea is that the vaccine was thought to be non-persistent, right, that cattle would clear, they wouldn't have a persistent infection. And what I'm suggesting from my work and from Cassandra's work is that those animals actually are infected and are competent to transmit, and that the vaccine could be providing a different kind of protection than they think. It could be providing a protection against acute disease because right, animals are immunized, but they also might provide an artificially induced endemic stability, which I thought was actually a really very positive aspect of the vaccine.
Well, but except that you may get ticks that could be then transmitting this to other hosts.
Yeah, and so I think this is the whole concept of dose. I think the transmission levels with these low levels of infection, it takes a lot of ticks to put that dose high enough to cause the severe disease. And that's why I think longicornis is so dangerous here, because, you know, we've all heard the stories about thousands of ticks. And, you know, we were able to transmit with 20 ticks, but what happens when you have 2,000 ticks?
But when you have low, so this dose dependence, early on you mentioned that so it sounded like it was dose dependence also in the severity of the disease. So you could get sick, but you were not gonna really have it, even for a horse.
So there's always the time courses involved too. The age of the animal is has a big impact on disease. So young animals, for some reason, are less susceptible to dying from disease. Also, animals that receive a low dose don't die from disease. But animals that receive a high dose or infected as naive adults very frequently progress to severe disease and death.
Yeah, it's 12 o'clock and I don't want to, but what you're saying is just all these parallels to the current pandemic you're experiencing with COVID, with how the vaccines are working. I mean, it's uncanny. So, you know, hey, One Health. So I want to let other people step forward, but also you're going to have 1 hour right now with the students, correct? So
Yes. Next by Francisco, he has a question.
Yeah, thank you, Glenn, for the talk. I really love all this Theileria, Babesia stuff. So yeah, it was very good to see some parasitology here at workers and in the seminar. I'm not the only one here that interested in also there's some other people. Yeah, so that's great. I was thinking about, I don't know, what is the epidemiology of our Babesia here in dogs in the US, because we know that the Asian Longhorn, they feed on dogs pretty well. And I don't know what is the epidemiology of Babesia in dogs, or Babesia canis or Vogeli, all the species complex, and also if there is anybody doing like vector competence for dog Babesia in those Asian Longhorn.
Yeah, so I think that's the answer to that question is it needs to be studied. Epidemiology in dogs is often associated with Rhipicephalus sanguineus, which is a tick that it's actually the only hard tick which can establish indoor populations in kennels. And so that's the most commonly accepted vector of babesiosis in dogs. But it would not surprise me if longicornis were to involve itself in that as well. And that just needs to be studied. And so the dogs is outside the area of the Animal Disease Research Institution out there in Pullman, but somebody needs to do that study, I think. And I don't know who will.
dogs are CDC people. So
Okay.
FYI, interesting question.
Okay. Yeah, thank you.
Yeah, I guess we have to stop here. If anybody wants to ask more questions, maybe you can stay for a few minutes at the student section. So thank you, Glenn, for the excellent seminar.
We'll be talking to you soon.
Yes.
Do I need to re-log in?